Hello everyone. Here I am going to tell about the herbarium techniques in this video. You all have a doubt what is meant by herbarium. Herbarium is a collection of dried specimens of a plant mounted on a herbarium sheets, identified and arranged sequentially according to the system of plant classification. If the plants could not be dried properly, they are maintained in a suitable preservatives. Bundle of herbarium species are stored in a small cabinet called herbarium case. Herbarium was first made by Cisalpini and his friends in 1550. The next one is uses of herbarium. Herbarium specimens show individual variation and evolution patterns of plants, so such studies may be conducted in the herbarium itself. Herbarium specimens are used as teaching aids. Herbarium provides specimens to match with the new plant to be described. Herbarium specimens are used for proposing new system of plant classification. Herbaria provide complete information about wild plants and their uses. There are some basic needs for plant collection. These are the things which we need during the plant collection: a chopper or a foldable knife, a pair of forceps, a trowel, a plastic bag, a label and tag, a vasculum, a pocket notebook and a pencil, a pocket lens which is used to observe the flowers, a wooden plant presser, drying sheets. Methods of plant collection. In the method of plant collection, the first information should be a collection of field data. Here, a field data means a collection of data of that plant which is present there in the field, whether it is a herb or shrub or climber or creeper, the color and smell of its flower, taste of its ripe or unripe fruit, regional name of a plant, utility of the plant for the local people, fluid secreted from the side of detachment of the plant. These are many information which should be collected in the collection of field data. The second information should be collected in the method of plant collection is collection of specimen. Frequently field trips are conducted to notice every stage of growth and reproduction. Specimens are collected usually at the time when they bear fruits and flowers. 2 or 3 specimens of suitable size are taken from a species, so we need not bother if one of the species are damaged. Fruits and seeds are usually collected separately. The collection number of specimens entered in a field notebook is given to the collected specimen. The next information should be collected in the method of plant collection is floristic study. All characters of collected specimen must be studied well in the laboratory. Then scientific name of the specimens are decided with the help of floras or by consulting experts. The next step is pressing of the specimen. After examining the plant specimens, the unwanted parts are trimmed off with scissors. Thereafter, they are pressed in between the drying sheet as earlier as the specimen look fresh. 1 or 2 drying sheets are spread on a flat ground and the leaves, flowers, fruits of that species are spread with the fingers so they have a natural appearance. The next step is drying. On the next day, the bundles are opened and wet sheets are carefully replaced by the new dry sheets. Again, they are tied into the bundles and exposed to the sunlight. During drying, the specimens are may be attacked by the fungal infection. To prevent that, DDT powder may be applied. For drying succulent plants, the drying sheets are changed twice or thrice a day until the specimens dry completely. The next step in the method of plant collection is poisoning of the specimen. The dried plant specimens may be damaged by small insect which feeds on the product. In order to protect the specimen from an insect, some poisoning agents are being added. They are mercury chloride solution is found to be a suitable poisoning for the plant specimens. In here, the mercury chloride is dissolved in alcohol and poured into the wide vessels. The specimens are kept dipped for 30 seconds and then dried between the drying sheets as usual. The next step in the method of plant collection is mounting of the specimen. After poisoning, the dried specimens are mounted on a thick whiteboard called herbarium sheet, which is thick as a postal card. They are prepared from a high quality paper. The specimen is placed on a paper on its upper surface. Gum is applied to its lower side using a brush. A drying sheet is placed over the specimen and some weight is kept over it for proper sticking. After drying, the drying sheets are removed. the herbarium sheets are examined if the gum has not stuck it properly. If so, little gum is applied to stick it properly. If this is not likely, strip of cellophane tapes are used to fix the specimen. If the plants are too small, they are put in a transparent envelope and the envelope itself is placed on the herbarium sheet. Flowers and fruits detached from the specimen during pressing and drying are put in the envelope and the envelope is placed on the same sheet in one corner. The next step in method of plant collection is labeling of the specimen. There is a printer label at the lower right corner of a herbarium sheet. All relevant information or collection of number entered in that label correctly. Now the herbarium sheet is the type specimen of the plant. There are some following information should be followed in the label. Labeling helps us to find out the effects of the habits on the morphology and distribution of plants. The next step in method of plant collection is arrangement of herbarium sheet. Prepared herbarium sheet are arranged in a specific order based on a standard system of classification. In India, Bentham and Hooker system of classification is adopted here most. Plants are arranged family-wise and an index card is prepared to indicate the exact location of this family. The next one is storage of herbarium sheet. The arranged herbarium sheet are maintained in special constructed herbarium case. The herbarium case is made from a reinforced steel with the help of welding works. Each pigeonhole contains a small gas-tight cabinet for holding herbarium sheet. Herbarium sheets of each and every family are placed in a cabinet. The family name is stick to each cabinet for easily identify the specimen. The next one is protection of herbarium sheet. Herbarium specimens are being eaten by herbarium beetles, drugstore beetles and etc. If they are left as such unmanaged, there may be a chance of losing of the specimen. So, cyanide gas, carbon disulfide gas, a mixture of ethylene dichloride and carbon tetrachloride or DTT is used to protect the herbarium specimen. The first 3 poisonous gases are pumped into the cabinet to kill insect and prevent the fungal growth. Thank you for watching this video.